The overall goal of this procedure is to capture cytosolic protein that bind to a specific sequence located in RNA molecule in mesothelioma cells.
This method can help answer key questions in RNA biology field, such as if a predicted RNA binding protein can bind adenosine-uridine-rich sequence in a transcript. The main advantage of this technique is that it complements functional characterization of stabilizing or destabilizing elements assessed by reporter assay. Demonstrating this technique will be Dr. Jelena Kresoja-Rakic, a postdoc from my laboratory.
To begin the protocol, obtain previously prepared ACC-MESO4 cells with an 80 to 90% confluence. Aspirate the medium, wash the cells with 15 milliliters of 1,X-phosphate buffered saline, or PBS, and aspirate the PBS. Next, add 3 milliliters of 0.25% trypsin EDTA and incubate the culture flask at 37 degrees Celsius for 3 minutes. Add 7 milliliters of supplemented RPMI 1640 medium, collect the cells in a 15 milliliter tube, and spin them down at 200 times G for 5 minutes. Then, aspirate the medium and resuspend the cell pellet with 1.5 milliliters of 1X PBS, then transfer the cells into a 1.5 milliliter microcentrifuge tube. Spin down the cells again at 500 times G and 4 degrees Celsius for 5 minutes. After the spin, discard the supernatant, resuspend the cell pellet with 1.5 milliliters of ice-cold 1X PBS, and spin down the cells at 500 times G and 4 degrees Celsius for